Hello everybody, good morning. My name is Rafaela. I'm from the BTA program. And today I'm going to talk about a little bit about genetic differentiation. Generally speaking, the genetic differentiation will lead from the appearance of new species. So let's see this image. Here you can see a lot of different birds, but they look like the same. In some kind you see some different beaks, and in other cases different feathers, but they are generally similar. Why they are similar? Because they belong to the same genus. They belong to the same genus, but they are different because they have different species. So how we have around the world so many different species from different animals, plants, bacteria, viruses, etc. Well, this process is called speciation. This speciation will happen in different processes. It can be allopatric, peripatric, or parapatric, or sympatric. The names are kind of weird, but the thing that happens, it's quite simple. So, first of all, you have three different groups of species, and these groups will start a different process that will lead to their differences. How it will happen? On the first case, for the allopatric one, you have these species having a barrier in the middle. So think about a hill, a big hill, and a group of animals equals live in that place, but in some point an earthquake happens. So you divide this group in two, and it's many years later, you will have these species from the that side being quite different from that one. Why it's happened? Because the things that happened here in this new hill will be different from that one. I don't know if scarcity of food, if in this hill has more water than that, if here happened a bacteria that kills a lot of animals here and not kill it in that side. Doesn't matter what affect, but the things that will affect the animals in that side will be different from this one. And based on natural selection, the animals in that side will be different from that one. For the peripatric or parapatric speciation, you have only the animals that was living in the same area. One look at each other and say, they don't like you anymore, I will move apart. And then they go for another side. And they start, don't talk each other. It's like a high school when the girls start to gossip and you don't talk with anybody. Yeah, so they start to move apart and move apart. And at some point, that group doesn't know everything that happened with this one. It's kind of a high school thing, okay? So they will become two different groups. And in the third one, I'm sorry, I didn't realize that it would be so clear, but here in the middle, you can start to see a yellowish part. This thing is just a mutation that happens in some kind of groups, and this mutation at some point will start to lead a different group inside that the same group. What will happen with all these differentiations? Sorry, okay, here is green and here is yellow. I should bring black and white. So in the end of the day, you will have different kind of individuals living together. So here you have one species, and here you have another one. Here you have one species, and here you have another one. And here inside this one, you have a new one appearing. And in the end, totally in the end of the day, you will see many different species appearing. This thing, for example, mainly for us in BTA, we are doing a lot of microbiology thing. And for example, let's say that you take E. coli bacteria and you create a colony, and you spread this colony in two different petri dishes, and one petri dish you put in an incubator at 37 degrees, and another one you leave on your bench. After a week, when you check these bacteria, they will be not so different. But let's say that you're repeating this process over a year. In the end of the year, probably the bacteria was grown on the incubator will be similar like every other, because they are in the same situation, but the bacteria who was living on the bench around 20 degrees will be different from that one. So you create two different kind of species. But let's say that you are doing this for 1,000 years. Then you have different species. And in some point, if you try to put them together, not for the bacteria because they have kind of different things, but let's say an animal. You have an animal living a condition here and another one here, and when you put them together, probably they will not be able to reproduce anymore because now they belong to different species. So that's the process, the speciation process that is led about the genetic differentiation. Okay? Thank you very much.